Okay, so I'll be sharing some of our work on building gene regulatory networks from single cell genomics, characterizing IL-10 producing memory T cells during aging. In the Meraldi lab, we're interested in understanding immune cell behavior in vivo. We want to know how genomic regulation contributes to different disease phenotypes. And we do this by modeling gene regulatory networks to learn the genes and transcription factors that are important to cell behavior. So in this study, we're characterizing regulation of immune response during aging. Young people are better able to control infection and mount robust immune responses to vaccines. And as we age, the immune system undergoes progressive dysfunction, often resulting in susceptibility to infection and poor response to vaccines. So our goal is to identify regulatory mechanisms contributing to this suppression of immune response with age. A well-known factor regulating immune response is the anti-inflammatory cytokine IL-10. IL-10 can be produced by a variety of immune cell types and suppresses a variety of immune responses. For example, deficiencies in IL-10 signaling have been linked to severe inflammatory bowel disease and asthma in humans. And IL-10 also plays an important role in preventing tissue damage during immune response to infection. It is known that IL-10 increases with age, combating chronic inflammation, but dampens immune responses. And less is known about the function of IL-10, its cellular source, or its regulation during aging. Recently, our collaborators in the Hildeman and Cunier labs at Cincinnati Children's examined immune cells in young and old mice in order to identify the cellular source of IL-10 during aging and determine its role in age-related immune suppression. They showed that systemic IL-10 increases in old mice. Much of this increase in IL-10 expression occurs in the spleen. And CD4 T cells were a major source of IL-10 in old mice. And then IL-10 expression is often associated with regulatory T cell function. Using FOXP3 GFP reporter mice, they showed that Tregs were not the major source of IL-10 in old mice, but it was the non-Treg FOXP3 negative CD4 memory T cells that had the highest capacity for IL-10 production. And then based on surface markers and certain cytokine dependencies, they showed that amongst the CD4 memory T cells, a TFH-like population were the dominant producers of IL-10 in old mice. So T follicular helper cells, or TFH cells, interact with B cells, playing an important role in their activation, selection, and differentiation into antibody-producing plasma cells. TFH function is critical for effective immunization. So our collaborators also showed that blocking IL-10 receptor during immunization of old mice restores vaccine response to levels similar to young mice. Thus, IL-10 limits B cell responses in old mice that are dependent on this TFH-like population. So we call this immunosuppressive age-associated IL-10 producing population TFH10. So the focus of our study is to characterize this mysterious TFH10 population using mathematical modeling of single cell genomics to address questions like how do TFH10 cells relate to kind of classic TFH how do TFH10 cells relate to classic TFH cells? How is gene regulation? Are genes regulated in TFH10? And why are TFH10 accumulating in old mice? And then ultimately, we want to know how we might re-engineer TFH10 behavior based on their knowledge of gene regulatory networks. So to answer these questions and characterize the entire CD4 memory T cell compartment, we use single-cell genomics to deeply profile T cells during aging. So we performed single-cell RNA-seq and single-cell attack-seq on CD4 memory T cells from spleen of young and old mice. And then using an IL10 GFP reporter mouse, we enriched for IL10-producing cells. We use these data in for a gene regulatory network. So later I'll describe how we interpret this network to describe regulation in different T cell subsets and age groups. So we clustered the single cell RNA-seq, and based on marker expression, we found 13 subsets of CD4 memory T cells, including TFH10 and what we're currently calling an intermediate TFH-like population. We clustered the single cell attack-seq separately and used label transfer from our single cell RNA-seq annotation, and we found clusters corresponding well to each of the T cell subsets. We hierarchically clustered the expression data and chromatin accessibility data at the pseudobulk level, and we found that the TFH10 population cluster closest to other TFH populations, confirming that TFH10's expression and accessibility profile is similar to that of TFH-like cells at genome scale. So here are the compositions of subsets in young and old cells for IL-10 positive and IL-10 negative populations. We found that of the IL-10 producing populations, we see a decrease in IL-10 positive Tregs in old age. And then we see an expansion of this TFH10 and a PRDM1 positive cytotoxic cell population in old age. Next, we set out to identify these age-associated IL-10 producing populations in published single-cell RNA-seq datasets of CD4 memory T cells. So we compared our dataset with 10 others, including aging cell atlases and a couple human datasets. So our dataset is the deepest profile of CD4 memory T cells with over 55,000 cells. And because of our IL-10 enrichment strategy, we capture many more cells from these age-associated populations. So Eliyahu et al. Is the next largest single-cell RNA-seq dataset of aging CD4 T cells. We annotated their cells and using our single-cell RNA-seq, and we map their cells to our UMAP embedding. And we see that our labels neatly separate their cells into more finely annotated populations. And we find evidence of all of our T cell subsets in their study. And we've also found evidence of TFH10 in human. So our TFH10 are similar to an immunosuppressive TFH-like population found in human tonsil. We've performed a similar analysis with many other data sets, and we find evidence of all of our populations, including TFH10 in many of them. And this kind of just shows that TFH10 isn't just some artifact of Cincinnati Children's Hospital. So we next wanted to characterize the gene regulatory networks of our T cell populations. We adapted a previously published GRN inference pipeline called the Inferolator, which models gene expression as a sparse linear combination of transcription factor activities. So I won't go into the details here, but I'll just point out a few pieces of the pipeline. So one of the inputs of GR inference is gene expression. So, and for single cell data, we input pseudobulk expression, where we combine counts for cells in the same population. And we found that this performs at least as well as using expression data at the single cell level. And we use the single cell attack-seq along with motif scanning to infer transcription factor binding. So we use this information to build a prior TF gene interaction network that helps kind of guide final network inference. So it's been shown previously that using chromatin accessibility to predict transcription factor binding improves GRN inference from gene expression data. And finally, we evaluate the quality of our networks using a gold standard network of interactions supported by experiments. We've curated many published TF-RNA-seq and ChIP-seq data sets in CD4 T cells to serve as our benchmarking data set. And that's helped us select optimal parameters. And then our final network uses these data as these data are incorporated into the prior for the final network. So we inferred a single gene regulatory network that describes all T cell subsets, young and old. And this network contains about 25,000 interactions, describing regulation of 2,500 gene targets by about 300 transcription factors. So our next challenge was how to interpret this network. How do we take a network of tens of thousands of interactions and find which parts are operating in different T cell subsets, different age groups? In particular, we want to know which interactions are operating in TFH10 and more specifically old TFH10 cells. So one way we do this is through core transcription factor analysis. So this identifies TFs that are responsible for the expression signature unique to each subset. So we first identify what we call signature genes. We find sets of genes that are either upregulated or downregulated in each subset. We also do this for, we also find age-related signature genes, which are basically differentially expressed genes between old and young within each subset. And then for each subset, we find TFs in our network with an enrichment of subset-specific signature genes. So this large heat map contains the core TFs for all 13 subsets. So a TF can either promote subset-specific gene expression in two ways. So red means that the TF activates gene expression for genes upregulated in that subset. And blue means that that TF represses genes downregulated in that subset. So here I've zoomed in on a piece of this. So for example, we found that BCL6, which is a well-known regulator in TFH cells, contributes to TFH gene expression as both an activator and repressor, which is consistent with the literature. We've also found that PRDM1, which encodes BLIMP1, is an important activator and repressor in these PRDM1 positive cytotoxic cells, as expected. So many of, and also many core TFs are shared across subsets, indicating partially overlapping modules of gene expression, gene regulation. So from this core TF analysis, we've extracted core networks for each T cell subset. So these networks contain core TFs regulating signature genes. So here I'm highlighting some of the interactions we predict to control genes important in TFH10. So squares are representing TFs, circles are target genes, the nodes are colored by relative gene expression across subsets, and the color of the arrows indicate the mode of regulation, so either activation or repression. And we see many interactions in our networks that are consistent with known CD4 T cell biology. So for example, we predict IL-10 expression in TFH10 to be regulated by PRDM1 and MAF. So these are validated regulators of IL-10. We also capture the mutual repression of TFs, PRDM1 and BCL6. These are well-known interactions controlling TFH versus TH1 differentiation. And then we also identify regulators of cytokine signaling that have been identified by our collaborators to be essential for TFH10 survival. So these include cytokine IL-21 and receptors for IL-6 signaling. We also define core networks describing regulation of genes that change with age. So here I'm showing TFH10 core TFs that regulate TFH10 age-dependent genes. Purple indicating higher gene expression in old, and green is higher in young. So of note, we find regulators of this age-dependent cytokine TNSF8, also known as CD153. This gene has been shown by several studies to increase with age in T cells. In one of those studies, CD153 is linked to age-dependent dysfunctional immune response and kidney injury, and it facilitates interaction with age-dependent B cells. So this is one of the single-cell RNA-seq data sets that we can compare to, and their dysfunctional age-associated population that expresses CD153 maps well to our TFH10 and intermediate TFH populations. So core TF gene regulation can be shared between T cell subsets. So we know that TFH10 are similar to TFH cells. So here I'm showing the overlap of core networks between TFH10 and TFH, and those that are unique to each subset. Shared between the two subsets are regulation of known TFH markers, as well as repression of cytotoxic genes. And then unique to TFH are well-known regulators BCL6, ASCL2, as well as genes important to TFH function, like CXCR5 and PD1. Unique to TFH10 are regulators PDRM1 and MAF, expression of immunosuppressive IL-10, CTLA4, LAC3, as well as pro-inflammatory interferon. So another thing we do with core network analysis is describe regulation of functional pathways. So when we perform gene set enrichment analysis on either subset-specific genes or relative gene expression across age, we found that pathways active in TFH10 were related to T cell migration and adhesion, TCR signaling, as well as T cell dysfunction. And we've identified regulation of genes associated with each of these pathways by core TFs associated with TFH10. You can see that most of the genes in these pathways and the TFs that regulate them are upregulated in old age. And many of these interactions could be contributing to the survival of TFH10 and its contribution to immune dysfunction in old age. So to summarize, so using single-cell genomics, we built a gene regulatory network describing CD4 T cells in youth and old age. We've identified core regulators of the age-associated immunosuppressive TFH10 population. And going forward, our goal is to experimentally perturb regulators that we've predicted to be important to the immunosuppressive function and survival of TFH10. So I'd like to thank members of the Meraldi lab, as well as our collaborators in the Hildeman and Cunier labs. And if you're interested in learning more about our work, check out Taryn's talk on Wednesday about TF binding prediction from AttackSeek. And also check out posters from Anthony and Alex on GRN inference in human cells. Thank you.